This is online lecture 19 for Bio 240, and we're going to continue our discussion of population genetics. So in the last lecture, we talked about populations that were in Hardy-Weinberg equilibrium, and we learned how to figure out the frequencies of genotypes and alleles within a population. Today, we're going to continue that discussion by talking about some of the important implications of populations in Hardy-Weinberg conditions. So let's start out by reviewing the conditions that are assumed to be true for a population that's in Hardy-Weinberg equilibrium. So first we have random mating between all the different genotypes. In other words, the phenotypic expression of a genotype doesn't influence mate selection in this population. So we have no new mutations. The third condition was no migration. So no individuals within that population leave the population and no new individuals come into the population. We have a large population. And we have no survival benefit for any genotype. And so if all of these conditions are true and a population is in Hardy-Weinberg equilibrium, there's a couple of important implications. First, this tells us that there will be no change in allelic and genotypic frequencies. From one generation to the next generation. And remember, our allelic frequencies were P and Q, and our genotypic frequencies were P squared, 2PQ, and Q squared. And the second implication for a population in Hardy-Weinberg equilibrium is that there will be no evolution. So evolution requires the selection of genotypes that are better suited to the environmental conditions, and evolution can only occur in populations that are not under Hardy-Weinberg conditions. In other words, there are new mutations, there is a survival benefit for some genotypes over others, and genotype and phenotype can influence mating. But of course, we know that all populations do evolve over time. So let's go through each of these conditions in turn and talk about what happens when those conditions are not met and how that leads to the process of evolution. So we'll start by talking about random mating. So if we do have random mating, any individual in a population has an equal chance of passing on its genes. So this means that the alleles in a population are going to be equally represented in the gametes of every individual in that population, which is going to go on to make the next generation. So there is random fertilization of gametes, and all of the alleles will be continued to represent in the same frequency. But as we know, there's not always random mating. So non-random mating can occur when we have inbreeding, and as you know, inbreeding is where related individuals preferentially mate. And we do this with dogs all the time. So dog breeders, plant breeders, a lot of different industries intentionally want to get purebred varieties and will inbreed them to make sure genetically and phenotypically homogeneous populations. In other words, inbreeding is going to lead to a decrease in genetic variance. And the reason this occurs is as a population becomes more and more inbred, the individuals that are inbred are going to have more homozygous loci than the random mating population. And some of the homozygous alleles are going to cause improved characteristics, but some of them will cause disease. And so that's a trade-off. For example, in dog breeding, by inbreeding populations, you can select for traits that are really desirable. For example, if you want a certain breed of dog to have one coat coloration and long fur, but that same population is also more at risk of other deleterious effects. So I know some populations of inbred dogs are at high risk of hip dysplasia or heart defects because of the inbreeding. And for this reason, in humans, we have both social taboos and laws against mating with related individuals, which does make sense genetically. Because as we saw when we did some of that pedigree analyses, related individuals, again, are more likely to be homozygous for certain recessive mutations that can cause really undesirable diseases. The opposite of inbreeding is outcrossing, where individuals within a population are specifically bred with unrelated individuals. And the point of this is to increase genetic diversity or genetic variance within a population. And genetically, this results in an increase in individuals that are heterozygous at many loci. And usually this leads to healthier populations and healthier individuals. And so we can describe this mathematically by using the inbreeding coefficient. Which is F. And it's a measure of the probability of having two alleles that came from a common ancestor. So the more inbred individuals are, the more likely they are to have alleles that came from a common ancestor, and therefore the higher their inbreeding coefficient. So if F equals 0, mating is completely random. Whereas if F equals 1, mating is related and the alleles are identical. Within a population that is inbreeding, there's going to be an increase in homozygosity, and so as part of that, we will get a decrease in heterozygosity. And remember, heterozygosity was 2pq. So remember, for our Hardy-Weinberg population, the frequency of a heterozygote genotype was 2pq. So we can express this decrease in heterozygosity by taking that frequency 2pq and multiplying that by our coefficient of inbreeding, f. And so this is the loss of heterozygosity within a population. So let's say we had a starting population with the genotypes AA, remember that is 2pq, big A, which was p squared, and little a, which was q squared. And with inbreeding, our heterozygous population, the big A, little a, is going to decrease by 2pq times f every generation. And that decreased frequency of the heterozygote is going to increase the frequency of the homozygotes. So our new frequencies in the next generation will be 2pq minus 2pqf for the heterozygote, and then for each homozygote we'll get p squared plus pqf and q squared plus pqf. So again, half of this decrease will go to each of the homozygote populations. And an important thing to note about this is that, as you can see, our genotypic frequencies are going to change. But the allelic frequencies stay the same. So we're just changing the amount of heterozygous with individuals within a population, but we're not changing how many dominant alleles or recessive alleles are within that population. So we're not changing the allelic frequency. Now let's look at our second condition for Hardy-Weinberg equilibrium, which is that there are no new mutations. Well, what happens if there is a mutation? So let's say our A allele, our dominant allele, acquires a mutation and it now becomes recessive. So if this occurs within our population, we're going to have a decrease in the frequency of P, and we'll have an increase in the frequency of Q. In other words, our allelic frequencies change if there are new mutations. In reality, mutations go in both directions. And some of these are even gain-of-function mutations. And in most populations, the mutation rate is very low. So for any given locus within 100 generations, you're unlikely to see any change going on. But over evolutionary time, it becomes very significant. So this condition, when not met, does change allelic frequency and does have a role in evolution. Our third condition for Hardy-Weinberg equilibrium is that there's no migration within the population. If there is migration either into or out of a population, migration has two consequences for a population. First, it prevents two populations from becoming genetically different from each other. Because they are, if you have two populations that are breeding within or breeding amongst each other, then you're going to keep mixing up the genetic information, and you won't get so divergent of information that you actually get a new species to form. And B, it increases genetic variation within a population, as migration into a population is going to bring in new alleles to that population. So for example, if you have a population that's A that has two alleles, and then a new individual comes into the population that has a different allele at that locus, now you've increased the genetic variation of the population. So if this condition, if no migration, is not met, again, increasing migration will change the allelic frequency within the population from one generation to the next. So this is also going to contribute to evolution. Now let's consider what happens if you don't have a large population. What if you just have a small population? Well, when this happens just by chance, same as with rolling the dice, by chance the alleles may not be represented in all of the gametes. So just a sample of the alleles will make it into the gametes of the next generation. And so, like when you toss a coin, it's possible that you can get skewed representation of the alleles in the next generation. And this is referred to as genetic drift. Where one allele is found at a higher frequency in the next generation than in the original population. For example, if you had two small populations, just by chance in one population, the frequency of the dominant allele for a given locus may increase, and by chance in the other population, it might decrease, and now you have a big difference between those two populations in the allelic frequency. And eventually it's possible for genetic drift to lead to one allele being completely lost from a population. For example, if you have an increase in the frequency of the dominant allele, and that goes on for a few generations, you could completely lose the recessive allele from that population. And this is called fixation. It means that all individuals in a population are homozygous for one allele. In other words, if P becomes 1 and Q is now 0, when we go to our genotypes, P squared is going to be 1 because 2PQ and Q squared are now 0, meaning that 100% of the population has the genotype of P squared, or the two dominant alleles. And the consequences of genetic drift are that you're going to have a change in allelic frequency within that population, the most extreme of which is fixation. You're also going to end up in, if we're talking about populations in a larger scale, you'll have increased variation between populations. So again, if one population drifts one direction and another population drifts the other direction, those two populations are becoming increasingly different from each other. And then within a population, you're going to have decreased variation. And that's because of this idea of fixation. So there's many consequences of this idea of genetic drift, all of which are important for evolution. And finally, let's look at the last condition, that there's no survival benefit for a given genotype. This is really just another way of saying that there's no natural selection occurring. So what if there is natural selection? Remember, natural selection occurs when one genotype produces more offspring than other genotypes. And this happens when a particular genotype is more successful in its environment than the other genotypes. We call this fitness, which is the relative reproductive success of a given genotype compared to the other genotypes. And we calculate fitness by measuring the mean number of offspring produced by one genotype divided by the mean number of offspring that's produced by the most successful genotype. So if the genotype you're looking at is the most successful, obviously the mean number over itself would be 1. So the maximum fitness is 1. So let's look at some numbers. Let's say we have the MN locus, and we're looking at offspring from individuals that are genotype MM, genotype MN, and genotype NN. So let's say we have 20 in this group, average of 15 offspring in this group, and an average of 10 offspring in this group. So which one is the most successful genotype? If you said MM, you're correct. So now pause the video here and see if you can figure out what is the fitness of each of these 3 genotypes in this population. Here are the correct answers. Remember, fitness is the mean, the average number of offspring for your genotype of interest over the most successful, the average number of offspring for the most successful. So for MM, it was the most successful is itself. So we take 20 over 20, and it has a maximum fitness. It has 1. For the MN genotype, they produce an average of 15 offspring. So we compare that to the fitness of the maximum group, which was 20, and you'll see that their fitness is 0.75. And for genotype NN, they produce an average number of 10 offspring. Compared that to the most successful group, which is 20, and you'll see that that gives them a fitness of 0.5. Sometimes we might want to calculate, rather than fitness, how strong the selection pressure is against a particular phenotype. And we call this the selection coefficient. So it's a measure of negative and this is calculated as 1 minus fitness. So for MM, the selective coefficient is 1 minus 1, which is 0. For MN, it would be 1 minus 0.75, 0.25. And for NN, it would be 1 minus 0.5, so 0.5. So again, these are the selection coefficient, and it tells us how much negative selective pressure there is for that genotype within the population in that environment. So the higher your fitness, the lower the selection coefficient, and vice versa. So of our 5 Hardy-Weinberg conditions, 4 of them, when not met, can lead to evolution or genetic change over time. Through changing allelic frequencies. And again, these were mutation, migration, natural selection, and genetic drift. Which is within small populations. And some of these evolutionary forces act in opposite directions, like migration and genetic drift, where migration increases genetic variation within a population, and genetic drift decreases genetic variation within a population. Natural selection can either increase or decrease variation within a population, and it can also increase or decrease the divergence between populations. So it's important to remember that real populations are affected by many of these evolutionary forces at the same time, and evolution is the outcome of all of these various forces and their selective pressures on the allelic frequency. So that's it for this lecture. I'll see you guys all in class.